Namaste friends, welcome back to Biology Center. In this video, we discuss about another important aspect from the genetics, that is chromosomal theory of inheritance. Chromosomal theory of inheritance. So before let ourselves into the outcomes of the theory, let us talk about the few important things about the Mendel, right? So already we know the Mendel did monohybrid experiments and dihybrid experiments for several years. Then he proposed and he published his research work. So everything was so we know. But so after immediately the finishing of the Mendel work, his work was not recognized for several years. So what was the reason? Actually, in the year of 1965. Mendel published his work, but his work remains unrecognized till 1900th year. So there are few reasons for unrecognition of the Mendel work till 1900. Yes, approximately there was a gap of several years. So let us find out the reasons for that. The first reasons for unrecognition of the Mendel work. The first reason is communication. Communication was not easy in those days, like so how we easily communicate with each other and how we easily disseminate the information to different parts of the world. That was not easy in those days. That was one reason. The second reason, usage of mathematical approach to explain the results. He was a mathematician, he was not a biologist, so he applied mathematical measures to explain biological concept. So other scientists, his contemporaries did not accept that. That was another reason. Next, third reason is a concept of genes as stable and discrete. So what Mendel said is, so genes are stable and they are distinct units. So they transmit from generation to generation. This concept was not much understood by other people. Next, fourth one, he could not so explain the continuous variation. Actually, continuous variation is in the nature. So one of his contemporary, Darwin, also busy in his research work. So they opposed some Mendel work because Mendel did not consider the continuous variation. So in his experiments, that was another reason. So one more one. So he could not give any physical proof for existence of genes, and he could not explain how genes were made of. So there was no physical proof for existence of genes. So Mendel said that so there are something going to be controlling the characters and transmitted from generation to generation. For that saying, he could not submit the physical proof. So these are few reasons responsible for unrecognition of the Mendel work till 1900. So after death of the Mendel, what happened? Then how Mendel work was recognized? So who are other scientists recognized the Mendel work and continued the work of Mendel? So in the year of 1900, three other scientists came into picture. Name of one person is Hugo De Vries. The name of the second person is Carl Correns. The third scientist was Van Schermark. Van Schermark. So they belongs to different countries. So they carry their research work independently. In the year of 19th year, these three scientists rediscovered the Mendel work. They focused on the results of the Mendel and do the experiments to know whether the results of Mendel are true or false. Right. Then, so in their time, so due to the advancement in microscope, so they observed the chromosomal movement. Chromosomal movement in meiosis. At the time of Mendel, so there was no advanced microscopes to observe these phenomena. So later on, so microscope technology was developed. So then other scientists were able to observe those structures present in the nucleus of the cell. So they are named as the chromosomes actually. The chroma means color, zoma means body. So here color bodies are named as the chromosomes. Right. Next, so after the discovering of the structures which are appear to be double and separate just before the cell division. So these structures, the double structures are called chromosomes. So after these three persons, so other two persons came into picture. So other scientists came into picture in the year of 1900 to two scientists or geneticists came into picture. So one person name is Walter Sutton. The name of the second person was Theodore Boveri. Two other scientists, Walter Sutton and Theodore Boveri, came into picture in the year of 1902 year. So they carried their research work independently. Right. Here, the Walter Sutton did his experiments on animal grasshopper. The experimental animal for his research was grasshopper. Then Theodore Boveri conducted experiments on the sea urchin. Sea urchin was the experimental animal for the Theodore Boveri experiments. So these two persons did experiments independently. Right. These two persons proposed the chromosomal theory of inheritance. Who proposed the chromosomal theory of inheritance? So Walter Sutton and Theodore Boveri. Popularly we can say that Sutton and Boveri proposed the chromosomal theory of inheritance. So let us see the outcomes of this theory. So what this theory says, what this theory explains. Simply this theory is meant for explaining the Mendel's principles or laws of Mendel. This theory says, chromosomal theory says, behavior of chromosomes is parallel to parallel to behavior of genes. How genes are behaving in pairing, separation and transmitting into the gametes, the same is followed by the chromosome also. That is why they said as behavior of chromosomes always parallel to the behavior of genes. So whatever Mendel explained, there he used the term genes. How the genes pair, so where the genes are located, how the genes are separated, how the genes sort into the gametes. So here these two persons used the term chromosomes. How the chromosomes paired, how the chromosomes separated, how the chromosomes segregated, how the chromosomes sort to the gametes. By using the structures chromosomes, Sutton and Boveri so wanted to explain the Mendel's laws. Next, before going to look at the observations made by these two persons, so it is better to know the comparison between the behavior of chromosomes and behavior of genes. Comparison between behavior of chromosomes and behavior of genes. Chromosomes always occurs in pair. Chromosomes always in a pair. Genes also always in pair. Occurs in pair. Second point. Separation of chromosomes occurs at the time of gamete formation. At the time of gamete formation. So here also same, the separation of genes, here separation of chromosome, that's it. Separation of genes occurs at the time of gamete formation. The third point about the chromosomes, independent pairs of chromosomes independently segregate of each other. And here a pair of genes separate independently of another pair. So almost these are similarities between the behavior of chromosomes and behavior of genes. So one more important point said by these two persons, genes present at specific locations on the homologous chromosomes, because chromosomes are the carriers of heredity. We know well, chromosomes are the carriers of heredity. So here, so I draw the chromosome, structure of chromosome. This is the chromosome. These are the chromatids. This is the centromere. So this is the locus. For example, locus. So on this locus, the particular gene is present. So homologous pair. If we draw the homologous pair, it is like this. So on this also, and at the same locus, these are the locus. At the same locus, genes are present. So in homologous pair, so one chromosome received from the father, that is called the another pair. So another chromosome received from the mother, paternal and maternal. The chromosome received from the father is called paternal. The chromosome received from the mother is called maternal chromosome. So let us move into the observations made by Sutton and Boveri. They made three main observations. Three main observations. Observations, right? The first observations, chromosomes like genes occur in a pair. So the first observation was chromosomes are in a pair. So how that pair make? This is the one chromosome. This is the another chromosome. Here genes present at specific locus. This set is called a homologous chromosome. This chromosome is received from the mother, called a maternal chromosome. This chromosome was received from the father, called a paternal chromosome. That was the first observation they made. So how here genes are in pair, right? How in pair? These chromosomes also present in a pair. The second observation, a pair of chromosomes separate at the time of gamete formation. So during the meiosis, so each pair separate and enters into the gametes. So this is the second observation. This observation was matched with the law of segregation principle given by the Mendel, right? So what observation made by Sutton and Boveri here? So matched with the principle of segregation or principle of purity of gametes principle. So let us see how. So this is the diploid cell in the G1 phase. In the G1 phase is called gap 1 phase or growth 1 phase. So here synthesis of DNA doesn't occur. Then the cell enters the S phase. So between G1 phase and G2 phase there is S phase. Here the knowledge of cell cycle and cell division is required. Then only it is easy to understand what I am going to explain now. So if you see the cell cycle and cell division, so mainly there are two phases. One is interphase, the another one is the mitotic phase. So interphase again divided into three phases. One is G1 phase, that is gap 1 phase. The second one is S phase, that is synthesis phase. The third one is G2 phase, this is gap 2 phase or growth 2 phase. So here we don't want to go in depth about this. Already we studied in the plus 11. So right, this is the diploid cell. In the S phase, the DNA synthesis, duplication of DNA strands will occur. The single strand become double. One DNA molecule become two DNA molecules. So here in the G2 phase, this is the condition of the cell which is in diploid condition. This cell is actually in diploid condition. So here the four pairs of chromosomes are very clearly appearing. So let us see how these chromosomes separated and enters into the different gametes. That is the important observation we are going to make now. So the cell which is in the G2 phase, so of course enters the meiosis 1 metaphase. Actually the meiosis also takes in two ways, meiosis 1 and meiosis 2. Under the meiosis, four phases are there, like prophase, metaphase, anaphase, telophase. So here, whatever phases are need to explain. So those phases only taken. So we skipped the other phases because it is the lengthy concept to explain here. That much of information was not needed. I hope you understood. Right. So how was the cell? How was this cell in the metaphase of the meiosis one here? So these 4 pairs of chromosomes already reached the equatorial place, right? This is the equatorial place. So all of them are almost in the middle of the cell. That is called metaphase, middle. Next, the same cell so enters the anaphase. So here the spindle fibers so already pulled out the chromosome pairs towards the poles. So this is the one pole, this is the one pole, this is the another pole, right? So two chromosomes so pull towards this pole, the remaining 2 chromosomes pull towards this pole. Then so after that cell division will occur, then 2 cells are formed. So in the meiosis 1, so what is the results of meiosis 1? The formation of 2 daughter cells from a parent cell. Next 2 daughter cells immediately enters the meiosis 2. Same thing so we are going to observe here. So how the 2 daughter cells in the metaphase of meiosis 2, they are like this. So here also chromosomes reached the middle of the cell, they are on the equatorial plate. So in case of anaphase, these chromosomes also pull towards the opposite poles. So in this cell and in this cell, then cell division occurs. So anaphase after the telophase, so then cell division occurs like this. Cell division occurs. Then 4 cells are formed. So here, so half of the chromosomes are reduced, and means one set of chromosomes only enters the gamete. These are the germ cells. Here, a diploid cell leads to the formation of 4 haploid cells during the meiosis. So this is the best examples for understanding the separation of chromosomes and so enters the gamete. The same thing was explained by Mendel by taking the factor, what we call them as genes now. Next, the third observation made by them. Chromosomes of different pairs, separate and assort independently to enter gametes. This observation was matched with the another Mendelian principle, that is law of independent assortment. Right? This third observation was matched with this principle. So here also we go for taking the diagrammatical representation. So by taking the example of Mendel example, R, capital R, capital Y, small y. So it is the plant in the heterozygous condition, that is a round shaped yellow color seed producing plant. This is a heterozygous diploid cell, a round yellow seed, responsible for round yellow seed producing plant. So here 2 possibilities are there. So according to this observation, so the members of different pairs so independently separate and assort independently so with any other chromosome. Here the any chromosome can pair with any other chromosome. So because of that reason, so we need to write the possibilities. Here 2 possibilities are written. The possibility 1. So before that you observe, so what are the chromosomes present in the diploid cell? The red color chromosome represents Y. So it is responsible for yellow color. The black color chromosome responsible for round shape. And small r chromosome is responsible for the this green color. The small y is responsible for the yellow. Right. So yellow, red, green, then wrinkled. In that way you have to understood. Next, possibility 1. In case of meiosis 1 of metaphase, this yellow chromosome can combine with the round shaped chromosome. Then this chromosome may have chance to combine with this chromosome. If this happens, the structure of the cell become like this. The chromosomes are arranged in the metaphase of meiosis 1 in this way. Next, so what happens in anaphase of meiosis 2? So almost these chromosomes, this set of chromosomes pull towards this pole. This set pull towards this pole. Next, so after meiosis 1, two daughter cells are formed. Then two daughter cells enters the meiosis 2. Then, so what is the condition of two daughter cells in the metaphase of the meiosis 2? So they are like this, right? So almost chromosomes are in the middle of the cell on the equatorial plate. Then in the anaphase of meiosis 2, the chromosomes are pulled towards the opposite pole in two daughter cells. Then ultimately from this cell, two haploid cells are formed. Totally here also 4 daughter cells, right? How many cells are formed here? 4 daughter cells are formed from this a single diploid cell. Here, so you have to observe how the chromosome pair separate and pair with other chromosome pair and enters the gamete. So this is the observation we need to make. Then come to the possibility 2. So in this case, in the possibility 1, this red chromosome the pair with this one, right? So here in this, so red may pair with this one, small r. Then blue with the black. The remaining process is same. So this cell enters the anaphase of meiosis 1. The chromosome sets are pulled towards the poles. Then in the metaphase of meiosis 2, the chromosomes on the equatorial plate. Then in the anaphase of meiosis 2, two daughter cells are formed. From each cell, daughter cell leads to the formation of two haploid cells. Here also, in the possibility 2 also, 4 daughter cells are formed from this diploid cell. So this is the example for understanding of the movement of chromosomes and pairing of chromosomes and how they enters into the gametes. Right. Finally, based on their observations, firstly, Sutton united the knowledge of chromosomal segregation with the Mendel's principles. So after their observations, they confirmed that. So what the Mendel said is right. Then Sutton wanted to unite the knowledge of chromosomal segregation with the Mendel's principles and called it as chromosomal theory of inheritance. So after uniting whatever knowledge he got from his own research experiment, so he called that as chromosomal theory of inheritance. The inheritance word taken from the Mendel principles of inheritance. Then the chromosomal theory was given by his own. So finally what these two persons, Sutton and Boveri, said. So here the pairing and separation of chromosomes leads to the segregation of factors, right? So how the chromosomes pair and separate, so in the same way genes also pair and separate then enters the gamete. This is the conclusion given by the Sutton and Boveri. So it is about a chromosomal theory of inheritance. Actually, after immediate proposal of this theory, it was put in controversy. So many other scientists were not ready to accept it, but so after the entry taken by the Thomas Hunt Morgan, so this was accepted. So T.H. Morgan, the father of experimental genetic, did experiments along with his students regarding this. Then the chromosome theory of inheritance was accepted. Thank you for watching this video.